Yeah, welcome back to class. We try to use this particular video to complete the topic on respiration, and for this class we shall be looking at cellular respiration, or tissue respiration as the name may be. Cellular respiration. So cellular respiration, as the name implies, must be a kind of process taking place in the cell. So that should be the first thing that you should have in mind. Cellular respiration is a kind of process that generates energy but taking place inside the cell, hence the cellular respiration. So in cellular respiration, we have two main processes that generate ATP, which is also the energy that we have mentioned. Two main processes that generate ATP. The two processes are, one, glycolysis, and the second one is Krebs cycle. So we try to look at these two processes briefly and where they occur and how they're able to generate ATP, just the very brief manner. Now let's start with glycolysis. Glycolysis. If you have followed some of our videos on in biology, you will understand the word lysis or lysis. The word lysis generally means breaking down. So glycolysis actually involves the breaking down of glucose. Breaking down of glucose to generate ATP. So glycolysis is the breaking down of glucose. Breaking down of glucose. Breaking down of glucose. Glucose to generate ATP, which is the energy currency of every organism. That is the energy used by every organism to carry out activities. So breaking down of glucose to generate ATP in the cell is referred to as glycolysis. Now, in the cell, glycolysis is actually a part of the cellular respiration, as we have mentioned. And this glycolysis occurs in the cytoplasm. It occurs in the cytoplasm. Okay? Occurs in the cytoplasm. At the end of glycolysis, energy in form of ATP is generated. Energy in form of ATP is generated. So this process called glycolysis actually involves some processes, and the major part I will mention in this glycolysis, which I will just have to draw something on the board, we have what we call phosphorylation. Phosphorylation of glucose. That is a very, very important one. Phosphorylation of glucose. Then we also have lysine, or breaking down. Breaking down of phosphorylated fructose. Phosphorylated fructose. Then the last one, which is the process that will generate the energy, and that is called oxidation. Oxidation of the 3-carbon sugar. oxidation of the 3-carbon sugar. Oxidation of the 3-carbon sugar. Now let's look at the diagrammatic expression of this glycolysis. That will give us a better view of what we are trying to explain in glycolysis. So the glycolysis process, we start with 6-carbon sugar that is known as glucose. So this is glucose. It's a 6-carbon sugar, 6-carbon sugar. 6-carbon sugar. This glucose will actually undergo what we call phosphorylation to give us what we call phosphorylated fructose. This process of phosphorylation is actually energy consuming process because phosphate group will have to be added to glucose to form what is called phosphorylated fructose. This process, which I refer to as phosphorylation, will involve 2 ATP. 2 ATP will be broken down to 2 ADP plus 2P. The 2P are the phosphate group that are derived from the breaking down of 2 molecules of ATP. And the process, which is actually referred to as phosphorylation, will end up giving us fructose biphosphates. Phosphate. In some cases, you just have phosphorylated fructose or phosphorylated fructose or fructose biphosphate or fructose diphosphate or phosphorylated 6-carbon sugar. We should not forget that fructose and glucose are isomers. They are isomers, compound having the same molecular formula but different structure. So we have phosphorylated fructose or 6-carbon sugar or fructose biphosphate. Now this particular process, which is known as phosphorylation of 6-carbon sugar, is actually energy consuming process. It's consuming energy. That is why we have these 2 molecules of ATP being broken down to 2 molecules of ADP and 2 phosphate group. It is this 2 phosphate group that will come to this fructose and convert it to phosphorylated fructose. Now from this phosphorylated fructose, we have the next event, which is the breaking down of the phosphorylated fructose to what we call pyruvate. The breaking down of phosphorylated fructose to 2 molecules of pyruvate. This is also 6-carbon sugar. 6-carbon sugar. Now the pyruvate are 3-carbon sugar. Pyruvate. It's 3-carbon sugar. We also have 3-carbon sugar here. Now this process of breaking down fructose to pyruvate is actually energy generating process. It's an energy generating process, and in the process each of the pyruvate will generate 2 ATP from each of these pyruvates, and we have 2 ATP being generated in the process of this splitting to pyruvate. So we have 2 ADP B plus 2P to give us 2 ATP. This other side also we have 2 ADP plus 2P to give us 2 ATP. That's what we're having. We're having 2 molecules of ATP being generated in that process. So at the end of this process, we have 4 ATPs. Now total ATPs generated is equal to 4 ATP. Why total ATP used is equal to 2 ATPs. Don't forget that these 2 ATPs were used in the process of phosphorylation process. So if 4 is generated at the end and we have made use of 2, that means the net ATP at the end of glycolysis is equals to 2 ATP. 2 ATPs. That will be the net ATP generated at the end of glycolysis. This process, as I've earlier said, occurs in the cytoplasm. Within the cell. Now, the second process, which is known as Krebs cycle. Krebs cycle. Somebody's name, so start with capital letter K. Krebs cycle. Krebs cycle is actually, in the real sense, we say it is a continuation of glycolysis, because the product of glycolysis will actually be used to to activate the process of Krebs cycle. Krebs cycle actually occurs in the mitochondrion. You have to keep that in mind, the mitochondrion. It occurs in the mitochondrion, and Krebs cycle actually involves the use of oxygen. It involves the use of oxygen. Now, Krebs cycle taking place in the mitochondrion will have to depend one way or the other on the product of glycolysis. And in some cases, where there is no pyruvate to form the acetyl-CoA, there will be a direct supply of 2 carbon compound from fatty acid. So in the case of Krebs cycle, why it is called a cycle, it is simply because the process is conservative and it undergoes a form of cycle, because the raw material that is actually used to initiate this particular process is recovered at the end of the process. The oxaloacetate that is actually used as a carrier to initiate the formation of citric acid is recovered at the end of the process. So it's a cycle. Now, how will this process start? Or how does this process commences? Now, the first thing we should keep in mind in this process is, once the process of glycolysis is completed, there is a double wall of mitochondrion. This pyruvate will start its journey straight into the mitochondrion, will start its journey straight into the mitochondrion, and this process will commence by this pyruvate losing one carbon atom, to form the carbon atom from the CO2. Now the pyruvate, which is 3 carbon compound, will become a 2 carbon compound, and this 2 carbon compound that is coming into this is actually the double membrane of the mitochondrion. This carbon compound that is being lost to form CO2. Now the 2 carbon compound, which is acetyl-CoA, it's a coenzyme, acetyl-CoA. This acetyl-CoA, this coenzyme, will come straight into, as 2 carbon compound, straight into the mitochondrion. In the mitochondrion, already waiting in the mitochondrion is a 4 carbon compound called oxaloacetate. So this acetyl-CoA combines with this oxaloacetate to form what we call citrate. Citrate, which is 6 carbon compound. Citrate, a 6 carbon compound. This oxaloacetate is actually readily available in the mitochondrion. The 6 carbon compound, which is known as citrate or citric acid, will start the cycle after the combination of the 2 carbon compound and the 4 carbon compound that is resident in the mitochondrion. That's why in some cases Krebs cycle could also be referred to as citric acid cycle. Now the citric acid will start undergoing some modification, and in the process we have other compound being formed. There's so many compounds being formed, for the sake of this class, we just mention few of them. Now this 6 carbon compound will metamorphose and lose a carbon compound to form what is called ketoglutarate. As a 5 carbon compound. And in this process, many ATP would have been formed. Ketoglutarate ATP would have been formed and released into the environment. And even between this citric acid and ketoglutarate, we have other compounds such as isocitrate, and many others being formed before the ATP is generated. This ketoglutarate acid or ketoglutarate later metamorphose to form other compounds such as malate or malic acid, and in the process CO2 is given off, ATP are also generated. In this process also we have CO2 also given off, since one carbon compound is actually released. Then this malate will later metamorphose. We have other compounds involved which are for compounds like fumate, acid, and so on. But just to mention few of them, we have this formalate, which is 4 carbon compound. 4 carbon compound. Then we have the oxaloacetate being recovered back. So these are some of the acid that are actually formed in the process of Krebs cycle. This ATP we are inserted in these different places, they are being formed as a result of some electron carrier And the major electron carriers that we have, they are NAD, nicotinamide adenine dinucleotide. Nicotinamide adenine dinucleotide. This nicotinamide adenine dinucleotide will actually will carry electron and become reduced NADH, that is reduced nicotinamide adenine dinucleotide. This is a good electron carrier. The electron is carried in form of hydrogen. Then we have other forms. We have another electron carrier, which is FAD in the process, and becomes FADH2. That is flavin adenine dinucleotide. These are electron carriers that activate the conversion of ADP to ATP. And so in all these places you are seeing ATP being formed, electron carriers have actually carried electron to form to convert the ADP available in the mitochondrion to be converted to ATP. Now, at the end of the process of Krebs cycle, don't forget I said the oxaloacetate is recovered back at the end of the process. At the end of this process, one thing you should notice, each of these pyruvates forming acetyl-CoA will generate 20 ATPs. So from these pyruvates down to the end of this process of recovering the oxaloacetate back, we have 20 ATP being generated. This other one will generate another 20 ATP. So coming together, 20 from each of them, from the process of glycolysis to the end of Krebs cycle, we have 40 ATPs being generated. But recall that the process of glycolysis always requires energy. 2 ATP being used. So at the end of the day, out of the 40 being generated as the total energy generated, 2 will be subtracted to pay for the 2 ATP used for phosphorylation. So at the end of a complete glycolysis and Krebs cycle, for the 2 pyruvates that are formed, we have 38 ATP being generated. 38 ATPs being generated. So we can say from the process of event now, we have only 2 from glycolysis and 36 from Krebs cycle, as the case as we want to put it. 2 from glycolysis, while we have 36 from Krebs cycle. After that will be the net. The net will now be 38 ATPs. Thank you very much.